We will now see in this video the C4 cycle. Already talking about the adaptations that are shown by the C4 plants, we have known that the two adaptations will be the dimorphic chloroplast present as well as the Kranz anatomy. So let us see how exactly the C4 cycle operates and prevents or reduces the loss of photorespiration. So let us consider the arrangement of the cells. This is the mesophyll cell present, and these are the bundle sheath cells. Okay, this is the mesophyll cell and the bundle sheath cells. Here there will be presence of the vascular bundle, here there will be epidermis present, right? So through the epidermis, what will be happening? Carbon dioxide will be entering into the mesophyll cell. Carbon dioxide from the atmosphere will first enter into these epidermal cells, and from the epidermal cells the carbon dioxide will enter into these mesophyll cells. In the mesophyll cells, as soon as the carbon dioxide enters, it is actually first going to react with water, and it is going to form a bicarbonate. How is this going to take place? Basically, there is an enzyme responsible for carrying out these reactions, which is called as the carbonic anhydrase. So what happens is, carbon dioxide in the mesophyll cell, carbon dioxide reacts with water to form H2CO3, okay, which is broken down into H plus and HCO3 minus by the enzyme carbonic anhydrase. This is the enzyme, the star sign shows it. Okay, so basically you can say that in the mesophyll cells, carbon dioxide is accepted in the form of bicarbonate, that is HCO3 minus. These a form of carbon dioxide which is entering into the mesophyll cell is now going to react with PEP. The PEP stands for phosphoenol pyruvate. Okay, this is a 3-carbon compound. Okay, phosphoenol pyruvate is a 3-carbon compound. It reacts with bicarbonate HCO3 minus, which is having 1 carbon. This both will react to form a 4-carbon acid, which is called as OAA. OAA stands for oxaloacetic acid. Okay, which is a 4-carbon compound found in the mesophyll cells. The enzyme that is responsible for these reactions is, the star mark there indicates, peptase. It stands for phosphoenol pyruvate carboxylase. That means it is always going to bind with carbon dioxide only. Rubisco is nowhere present in the mesophyll cells. These oxaloacetic acid is the first product of the C4 cycle, and as a result, this cycle is also called as a C4 cycle, and the plants are called as the C4 plants because the first product form is that of a 4-carbon compound. Now, these oxaloacetic acid which is formed in the mesophyll cell will convert itself either into malic acid or aspartic acid. Now, where is this going to be formed? If there is no ammonia available, oxaloacetic acid is going to convert itself into another 4-carbon compound called as malic acid. Or else, if there is ammonia available, NH2, amino group available, then these OA is going to convert itself into aspartic acid. Okay, remember, oxaloacetic acid is going to convert itself into another 4-carbon acid, which can either be malic acid or aspartic acid. If amino group is available, then it is going to convert itself into aspartic acid. Either of the two, whichever is the compound formed, will transfer itself into the nearby cell, that is the bundle sheath cell. This is nothing but the plasmodesmata. Through these plasmodesmata, the connection that is present between the two cells, the aspartic acid or malic acid from the mesophyll cell will now enter into the bundle sheath cell. So let us consider here malic acid entering into the bundle sheath cell. Again, as I said, malic acid is a 4-carbon compound. Here it will undergo the process of decarboxylation. Here, when carbon dioxide is fixed, fixation is taking place, which will also be called as carboxylation process, like that we had seen in the C3 cycle. Once the malic acid is entering into the bundle sheath cell, here it will again break down to form a 1-carbon compound and a 3-carbon compound. The 1-carbon compound that is released is none other than the carbon dioxide, whereas the 3-carbon compound formed is PEP. Instead of PEP, actually the one which is formed here is pyruvate or pyruvic acid. These carbon dioxide which is released in the bundle sheath cell will now combine with RUBP in the presence of rubisco. Remember, we have just discussed in the earlier video that mesophyll cells, they have a granule chloroplast where there is no rubisco present, whereas the bundle sheath cells, they have a granule chloroplast where the rubisco enzyme is present. So here basically what is going to take place is the C3 cycle. So basically, whether you consider C3 plants or C4 plants, Kelvin cycle or the C3 cycle is going to take place. Only the site where the cycle takes place is different. In C3 plants, the Kelvin cycle takes place in the mesophyll cells, whereas in C4 plants, the Kelvin cycle takes place in the bundle sheath cell. These pyruvate, which is actually formed here in the bundle sheath cell, will now be again transported back into the mesophyll cell, where it is again going to react with these bicarbonate to form next 4-carbon acid, that is oxaloacetic acid. And while it is transported here, ATP will be utilized to form adenosine monophosphate. Okay. So it has all the benefits, only one thing is there will be energy expenditure here taking place for the C4 plants. It is a bit, C4 cycle is a bit expensive, okay, as compared to the C3 cycle. So let us again quickly revise it. In C4 plants, the mesophyll and bundle sheath cells are present. The mesophyll cells will accept the carbon dioxide in the form of a bicarbonate, which reacts with a 3-carbon compound called as phosphoenol pyruvate. The enzyme peptase or phosphoenol pyruvate carboxylase is going to form the first product of the C4 plant, the C4 cycle, that is oxaloacetic acid. These oxaloacetic acid is either converted into malic acid or aspartic acid, which will be transported into the bundle sheath cells through the plasmodesmata, where it is the process of, you can say, decarboxylation is taking place. Right, because carbon dioxide is released. So this decarboxylation process is resulting into the Kelvin cycle in the bundle sheath cell, and the 3-carbon acid formed is transferred back into the mesophyll cell. So this is the way in which, as you can see here, there is no rubisco present. There will be no photorespiration taking place. Here there is rubisco present, but there is no oxygen available in the bundle sheet cell because non-cyclic photophosphorylation is going to take place here. High temperature will be tolerated by the mesophyll cells. Oxygen is released in the mesophyll cells where there is no rubisco present. So rubisco in either of the case cannot act as oxygenase because of its absence. Where it is present, there is no oxygen available and the temperature is also low in the inner cells as compared to the outer cells. So here the rubisco is always going to behave as carboxylase and the Kelvin cycle is going to be continued to form the glucose molecule. Like the C3 cycle was requiring 6 cycles to form one glucose, similarly the C4 cycle also have to be carried out 6 times to form the one glucose molecule. Why? Because 6 times the cycle takes place, 6 carbon molecules will be released, carbon dioxide, and those 6 carbon dioxide will be entering into the Kelvin cycle to form the one glucose molecule. This is all about your C4 plants and C3 plants. Thank you very much.